Okay, students. So now let us start the process of embryo formation. What is embryogeny? The complete process starting from the zygote that result in the formation of a complete embryo is known as embryogeny. The process starts from a single cell structure that we say zygote. How is this zygote formed? We have done that zygote is formed as a result of syngamy, that is fusion between the male and the female nuclei gamete. So this zygote is 2N structure. We have done that the endosperm is 3N structure, zygote is 2N structure. Now this will divide by the mitosis division, and this is unequal division. The upper one is a smaller and the lower one is the bigger. You can draw vice versa also, there is no issue about it. This bigger one is known as the suspensor cell. And the upper one is the going to form embryo. Now this suspensor cell will divide again by few division and it forms a 6 to 8 structure. The lowermost part of this structure gets swollen and this is called haustoria. By this haustoria, this gets attached to the embryo cell. It is attached to the embryo cell with the help of this haustoria. And this uppermost cell, this uppermost cell is called hypophysis. This hypophysis is going to form radicle and the root cap. Now see this part. This cell divide by 2 vertical division. Vertical division means like one division is like this and second is like this. And one transverse condition like this. So as a result of 2 vertical and 1 transverse division, 8 cell structure is formed. Now first of all, this is going to form a globular embryo. A globular embryo is formed. Now the cells of this globular embryo, they arrange themselves into an outer layer that I have shown you with the help of this green color and the inner cell mass. This outer layer, this is called dermatogen. And this inner one is called procambium. This dermatogen, this is going to form the epidermis. And procambium, this is going to form the ground tissue. Do you remember ground tissue kaun se hote hain? It is cortex and pith plus vascular tissue. So is that clear? That globular embryo, this globular embryo will give rise to an outer layer that is called dermatogen and the inner cell mass that is called procambium. From procambium, ground tissue and vascular tissues are formed. Now from this globular embryo, the shape of the embryo becomes heart shape. So this is called heart shape embryo here. And ultimately, in this heart-shaped embryo, now see the changes, and this is going to form this mature embryo in which cotyledon appear. These are cotyledons, and this whole structure, this is called embryonal axis. Have you noticed that in the beginning, the size of these cells is used to be bigger, but in the final stages, their size becomes shorter and the whole space is now occupied by the embryo. Students, these different stages of embryo formation, these are quite important from the examination point of view. So you have to do the practice of the drawing also.